So hi, everyone. Welcome. Thank you, Sharon. Thank you, WHN, for organizing this. Exciting to talk with you today. I think we're going to do something very informal. I think the idea is that, there's a lot of beeping, but it's fine. We're going to do something very informal. I think the idea is that Dave and I are going to do some very short presentation. I'm going to try to stick with 20 minutes, my talk, but I'm going to show a lot of photos. And I think Dave's going to probably present around 20 minutes as well. And then we're going to open for some questions. For those who don't know, as Sharon said, I'm a neuroscientist. I'm going to talk a little bit about my research, as well as some of the research that is going on in neuroCOVID right now. And I think David's going to come more, how he said, from the bench to the bedside. David is a clinician, so he has the clinical aspect of COVID, neuroCOVID, long COVID, and I'm going to cover more of the basic science. And yeah, let's start. So I'm going to do about a 20-minute presentation, and then Dave's going to do a 20-minute presentation too, I think. So let me share my slides so we can start. And I'm going to just close the video so it's don't freeze my computer, but I'll be back after it. Okay, let me just share here. Okay, can you see my screen?
Oh, good.
Okay, perfect. Thank you. So I will try to stick with the 20 minutes. I have a lot of data. I tried to do a presentation. I was kind of updating the presentation all the time. So it's a bit, I tried to stick with a kind of organized flow. It's a bit confusing. I hope it's not too confusing. But for those who don't know me, I work at the California National Primate Research Center, and I do a lot of microscopy. I'm basically mostly a microscopist. And this is one of the photos I do in the microscopes. We're kind of looking how glial cells, so cells in the brain, are reacting to the presence of SARS-CoV in the brain. I always like to make start with an introduction just to present myself. I am actually, as I said, in the Primate Center. I work with monkeys, so this disclaimer, I know it's a bit uncomfortable for some people, but I think it's important to highlight that some of the data I will be presenting today was performed with monkeys, and I work with one of the 7 national primate research centers here in the US. I'm in California. You can see in Northern California. There are 7 spread around the country here. Despite having these primate centers, and we have around 5,000 rhesus macaques in our primate center now at California, it's very, very little research is conducted in monkeys in health science, basically. So it's just around 0.5%. And they are very important because it's if you think about the COVID vaccines, or if you think about other very translational things we do that are necessary for human health, they're being done in primates. So it's kind of required. I think it's important for some type of research is very crucial. It's not everything, but some type, especially neuroscience and things that look for cognition and behavior that is really hard to translate in rodent models. And one of the things we're very proud at our primate center is that we have a geriatric colony. So we have our monkeys age around 3 times faster than humans. So we kind of have these animals that we are expanding our geriatric colony. I am particularly interested in the neurodegeneration, aging, and Alzheimer, and how this can correlate with viral infections as well. And I'm going to show a little bit of data on this overlap between Alzheimer pathology and COVID in the end, close to the end of the presentation. And just to summarize what we do here is we're not only interested in COVID. COVID has been one of the major goals of my research now, but we are also interested in other viral infections. So this is first in the top here. I don't know if you can see the cursor, but this is the research we do. We go from cognitive evaluation with the monkeys to in vivo imaging to a lot of microscopy. We are basically a microscopy lab, and we do high resolution microscopy. So going from tens of microns to nanomolar, so very, very basically just looking at how one neuron it's interacting with another. And some of the things we develop in our primate center is just some monkey models for Zika infection. So like this, like, and it's like all these viruses. So this is Zika, SARS-CoV here in the middle, and HIV. HIV is a bit different because the monkey has a different viruses, which is SIV, which is the simian version. But we kind of like have trying to develop these monkey models of pathologies that are very translationally close to what we see in humans. And this is especially relevant for SARS-CoV because SARS-CoV, it's not, the virus doesn't bind to the same way that it binds from rodents to humans. So like you need to have a genetic modification in the rodent because the virus, the spike protein of SARS-CoV-2 doesn't bind ACE2 receptor from rodents normally. So, but it binds to monkeys. So that's why we can have like a very, very similar pathology, SARS-CoV infection pathology. We see in monkeys, we can have we can see in humans, we can have in monkeys as well. And just moving on about like this general idea of what it is, like brain pathology and viral infection in the brain. Like it's surprising as someone that study neuroscience that there's still a lot of resistance with the idea that the viruses can directly enter and affect the brain. Because if you look at the history of all like the other types of viruses, and you can go from DNA, and here in this table in the right, you can look at DNA viruses such as herpes virus or JC virus. We know they infect, like directly infect neuronal cells. And here you can see different types of cells. You can see a lot of the viruses infect neurons, but we also have different cell types in the brain, like oligodendrocytes, which are like represented here in this image. So if you have a neuron here in orange, oligodendrocyte will be the cell in blue. We have astrocytes, which are here, another cell type in green. And we have microglia, which you're gonna be hearing more soon because it's my favorite cell type, and it's the immune cells of the brain, microglia, and you can see how they're like working together here with the neurons and with the other cell types. And this is kind of like the major, there are more cell types in the brain, but these are kind of like the major cells we see. And you can see that basically a lot of these cell types can get infected with different viruses. So we have like HIV, for example, can infect microglia. You have a lot of viruses, a lot of the RNA viruses can infect neurons. So this is well known. So the fact that we have a virus such as SARS-CoV infecting cells in the brain, it shouldn't be a surprise because we know it comes from other viruses as well. And as I said, microglia is the type of cell that I'm mostly interested in because they are really the immune cells of the brain, and they have a special relationship with neurons that and this is like the joke here is like, you have a neuron, you have a microglia talking to a neuron, I'm gonna eat your synapses, like just the ones I'm not using, right? The ones I'm not using. Because this is one of the things we see in inflammation in the brain is that the relationship between neurons and microglia, it changes. These cells, the microglia get activated and it kind of like starts eating synapses, like this connection between neurons, even from neurons that are healthy, because it shifts in a profile. So this is one of the things we say it's a neuroinflammatory response. And one of the things I've used microscopy for is just see how microglia, and here in red in this image below, and neurons in blue, how they are interacting. So kind of like I can do some 3D analysis to see how the microglia is eating the synapses to the neurons. It's like, is this healthy? Are these cells inducing neurons to die? So these are some of the things I'm doing in animals infected with COVID. Okay, and talking about, okay, you have like these different cell types and you have different viruses coming to the brain. So what is about coronaviruses? And I'm not even saying SARS-CoV-2, I'm just saying coronaviruses in general, why they are so dangerous for the brain. And more than other different types of viruses that I mentioned earlier, because what happens with coronaviruses is that they can really enter the brain through different routes. So like they not only have one mechanism that they can access the special. We like to think that the brain is isolated and special, but it's not so much. We don't have anything so isolated in the brain. The brain is all connected, in the body is all connected. The brain is part of the body, is also connected. So basically you have like, we know, and I'm not going into details with the science, but it's just like at least 4 different routes that we know that coronaviruses can hijack the system and enter the brain. So it's like direct viral infection through olfactory nerves, which will be the A. but also like other routes involving blood vessels. So this will be a blood vessel. So basically viruses such as SARS-CoV-2, SARS-CoV-1, MERS, all the coronaviruses we know can enter the brain through like opening the walls of the blood vessels or hijacking immune cells or like really like disrupting how these cells are connected to each other. And the thing about coronavirus is that we know like some flu strains, so some different like H1 or HB, like different types of influenza, they can do that, but usually one or another mechanism. So really what happens is that you ended up not having so much viruses in the brain. Like having so much viral infection in the brain, it's very particularly to some types of viruses that are able to do all this type of entry. With an exception, and this is not the topic of today's talk, but I just want to highlight that H5N1, actually the H5NX series, it's very unique. It's not your type of influenza, and it's concerning because this is like the bird flu is H5N1 and can be very neurotropic as well. So it's more neurotropic than the normal influenza viruses. And we scientists know that actually H5N1 can enter and can spread through multiple regions in the body, in the brain. So it's not like really like your traditional influenza virus, but most influenza viruses cannot do that. So it's rare to find them in the brain. And not surprising, like when we saw that there is, okay, coronaviruses and you have a pandemic that there are so many million infections worldwide, like we are going to have a lot of cases of the virus coming to the brain. And not surprising, this was kind of like scientists have been seeing over those last 5 years. I always like to put this study, mention this study, which was from 2022, which is one of the landmarks and just showing, and it's the perfect study because they have MRI from patients. They were looking at the brain of people before and after infection. So they have a lot of participants, which is like a really important study. And what they saw is not surprising, like the viruses was really entering the brain. So like comparing patients with infection and a lot of patients without infection. So a before and after, basically, which was great because each patient was basically their own controls, right? Before and after infection. And what they saw in this study is that there is a lot of really big morphological changes in the brain. So you have like basically a lot of the things we see with neurodegenerative processes like Alzheimer, they're kind of like happening in the brain of these patients after a few months of COVID infection. So it's a lot of regions here, but basically you have like a shrinkage of some of the brain areas that are responsible for memory and cognition. And you have like a lot of these regions that I'm going to mention with microscopy are some of the regions they saw like neuronal loss. So it's neural, like when we say atrophy and shrinking is really like neuronal cells dying mostly. And what they also saw is that there is like a change in the thickness of some cortical regions. And again, changing of the thickness, enlargement, it means tissue dying, like you have inflammation, you have neurons dying. So you're kind of like having this loss. And when we compare with and they were looking here at the orbitofrontal cortex before and after control. So control, you have a normal variation here of 1%, but you can see here with the cases, you go up to 2% of change, especially with older patients. So there is a change like in how this like behavior changes, like when we compare behavior with thickness of the cortex. So there is a cognitive impairment that is coming with the brain regions changing, really. And the orbitofrontal cortex, I highlight because it's exactly one of the regions they saw a change in the patients. Is also one of the regions I see change in microscopy. So the orbitofrontal cortex, and that's one of the things. So like if we imagine that the virus is coming through the nose, so when we have like an airborne virus such as SARS-CoV, the orbitofrontal cortex is like it's a complicated name, but basically it's just a part of the prefrontal cortex, which is also secondary olfactory cortex. So it can get the virus really fast if it enters the brain in a few hours. But it's also a region that is one of our most precious regions in the brain. It's very special because it's one of these regions that like it gets fully developed very late, like when we are usually in our mid to late 20s. So it's like, so it's concerning for people like kids and adolescents that don't have this region fully developed yet. And it's also like what we see in the microscope in this image here in the right is that, like, remember I mentioned we have different cell types in the brain. So these are astrocytes are one of them in red, and we have neurons here in green. And we kind of like here, we have the cortical layers. And what we see in infections cases is like there is this leakage. So we see basically there is like a gray matter will be here, and white matter will be here. We kind of like have a shift from the cells in the white matter invading the gray matter, which is basically you have all these astrocytes like attacking the neurons, and these neurons are going to be dying. So like you have this inflammatory response that induce neuron loss. And we're seeing this in the orbitofrontal cortex. So it's an olfactory region, but these neurons are not only responsible for olfaction, they are also involved in inhibitory behavior. So they are like, and it's kind of like very high functional neurons that we don't want to lose. So it's like, that's the thing, like it's mostly concerned, like people need to realize that the olfactory cortex doesn't mean that these neurons in the olfactory cortex are only taking care of olfaction. They are also taking care of our personality, of like how we behave, like how we process emotions. So these are all combined in the brain. There's not like an isolated region for each type of feeling or each type of function. Another thing, so talking like exactly about the research we do in our primate center. So we are developing a monkey model of COVID, because as I mentioned, like similar to humans, the monkeys can have a respiratory pathology. So they have some monkeys, they have mild symptoms. Some monkeys, they have very aggressive symptoms, just like humans. And one of the things we were doing is just trying to see, and that's exactly like how I ended up in this project. It was just like, can we see neuronal infection? And actually, yes. So we published in 2022 this paper just showing like when we have young and aged monkeys infected with SARS-CoV, and we see in both. So not only in aged monkeys, but we see pathology in the young monkeys as well. So here it will be like just a video showing a neuron. Like I don't want to get too complicated again, but I think the important message here is that we have a neuron that is infected with SARS-CoV-2. And not only we see spike protein, which will be in purple, but we also see replicating virus in the cell body of the neuron. So it's really like it's causing like these neurons basically in the process of dying. And we know that because the nuclei is fragmented. So if you see the nuclei here is a 3D reconstruction. And if you see the nuclei in blue, you see these cells around, which are non-neurons. They are intact. And you see the nuclei of the neuron here in the middle is fragmented. So this neuron, like we caught a neuron in the process of dying, and we can analyze that by doing this type of 3D reconstruction. And this will be like an image summarizing a lot of the things I see. So a lot of people say, oh, so might be the vaccine. How are you sure that the spike protein of the vaccine is not doing a similar thing of the COVID infection? It is not the same thing, because I don't see only spike protein. I do see spike protein, but I also see active viral replication. And active viral replication inside a neuron cannot be caused by a vaccine, because a vaccine is just fragments. It's just spike protein, or it's not like you cannot induce an active viral replication inside a neuron by the vaccine. And also I see nucleocapsid. We also see other viral proteins as well. But it's like one of the major things, like not only the virus is entering the brain, but it's actually replicating and preferentially neurons. And this is one of the things we see. And so we published this in 2022. I'm not going to go into the details of all the science and quantification because it's boring, but it's just the ideas that we saw that in a few days, we are able to see not only the viruses is going to primary olfactory cortex regions here in green. I'm going to go with the complicated names, but we go to the secondary cortex and we go to the tertiary cortex. And one of the concerns here is that, and we were quantifying how much virus, so we can quantify exactly how much virus it is in each region, and how in compared to young and versus aged animals. So we do see more pathology in aged animals, but the pathology was also present in young animals. So young animals, and it's exactly what we see in humans too, like patients. Aged patients sometimes have more serious complications after COVID, but that doesn't mean that younger people are not having complications as well. And these brain regions, as I said, they are involved in many other functions. They're not only olfactory, they're like we have here hippocampus, entorhinal cortex, which is one of ERC here, entorhinal cortex, which is one of the regions we know like are involved in Alzheimer disease beginning. So these are all regions that are relevant for memory and cognition, and they also are part of the olfactory cortex. And going very science here very briefly, but I think one of the alarming things for me and for other scientists as well was see the active viral replication inside neurons in the brain. And like, and this is something like the literature, this is a paper that came out even before the pandemic. It's just showing like how the virus can change the structure of cells to allow active viral replication. So you basically have like these compartments inside the cell that are causing, like, it's just like genomic RNA. So it's basically just like the virus is really creating like an incubator of virus inside other cells. And this is exactly what I see in neurons as well. So you can see here, this like will be green protein is neuronal protein, nuclei of all cells is blue, and red is a viral protein. So we see active viral protein shifting the neuronal protein. So again, this is another neuron that is dying. So you can quantify how much protein. This is all like baby virus, like ready to be released when this neuron collapse, and it's just going to infect other neurons. So just seeing this in the microscope, I never seen anything like that, because Zika virus and HIV virus, it did not cause this type of damage that I see with SARS-CoV. But moving on, a lot of the things, clinical data is coming as well, is the cerebral hemorrhages. So a lot of patients like increasing this idea that you can have increase in stroke, increasing this idea that you have a disruption of the blood vessels. And again, if you think about that, the virus can hijack blood vessels that are coming to the brain to enter the brain tissue. So it's not surprising that you're seeing all the like this blood vessel disruptions. So these are some of the one we see, and this is how they look like in the microscope. You see like this large blood vessels. Green is neurons. So you kind of like see like that basically these large blood vessels, and red, it's kind of like disrupting the neurons that are surrounding here, shows the same, like how you have like this leakage, like it's coming from the gray matter, it's coming from the white matter to the gray matter, and it's just like causing a lot of inflammation and glial, what we call like astrocyte and glial activation, which are the other cell types, as I said. And we can quantify like how disrupted are these blood vessels in the brain. So some of the things I do is like just isolate some of these blood vessels arriving to the brain tissue and see how they look like. And structurally, when we see in the microscope, they are very disrupted. Like you see the collagen, so like the walls of the vessels are compromised. And not only they're compromised, but we see presence of viral protein inside the blood vessels. And it's kind of like you can see here in a 3D reconstruction of a piece of blood vessel coming to the brain of the infected animals, and we basically see a lot of the here compromise in the wall of the blood vessel, and we see all this virus accumulating. So it's very clear, and we can quantify all this pathology. It's not that hard to quantify it. And all of this, while I have been seeing, and other scientists and neuroscientists are seeing, is just like there is this increase in cases of neurological disease associated with COVID. So I like to highlight this work from a group from UCSF that I think is a really, really great, it's a review, but it basically shows like how this brain vascular and this idea that the neuroscience is changing, really, like the field we are changing in terms of like, we're realizing that the brain is not so protected and isolated as we thought it was, as we wish it was. Actually, the brain is very connected. So you have all these factors that are contributing to disrupt like the blood vessels in the brain, and are contributing to inflammation. And I like this review because it puts COVID-19 as a neurological disease here together, like side by side with so many more known diseases such as Alzheimer, multiple sclerosis, stroke, traumatic brain injury. Everybody heard that these conditions cause neurological damage and disease, but we don't hear so much of COVID. But it's like neuroscience field and people that are working in neurovirology are putting COVID-19 as a neurological disease, exactly because it's causing this disruption of blood vessels, and it's compromising how the blood it's coming to the brain, and what else is coming together with the red blood cells, right? And we need like this idea that the field is changing, and we need a more integrated approach. So we not only need neuroscientists, but we need immunologists, hematologists, imaging, like other areas, like working together to understand what is actually happening in the brain of patients with COVID. And again, like this is another review. I think it's very interesting, just showing how like this blood-brain disruption, it's a major cause of probably it's a major cause of complications in patients that have neurological symptoms after the COVID infection, like they're developing long COVID. And this study, I think, is really interesting that came out last year because it kind of like shows a different technique, a new technique. And it's basically just imaging the brains and just showing like the regions that they're seeing a difference in volume and difference of thickness of the tissue. So the tissue is reducing, is disappearing, is exactly the same regions I show the changes in the microscope. So it's the orbitofrontal cortex here in the front, which is the secondary olfactory cortex, is here, the piriform, the entorhinal cortex, which is primary olfactory cortex. So basically the same regions I see the virus in the microscope are also the same regions that the patients are showing alterations in brain imaging in scan. So it's not a coincidence that this virus is appearing in these regions. It is the same regions that are showing changes for patients as well. And yeah, and this idea that will be like these three major things we see that can be caused by the SARS-CoV, which is like you can have direct viral infection, you can have disruption of the blood-brain barrier, and you can have neuroinflammation. You can have the three of them together, they overlap, they contribute to each other, but you can also have them separately. You can have people that are only going to have direct viral infection without a lot of inflammation, but this is enough to cause a lot of problems, we know, and these are some of the problems that we are associating now with presence of these factors in the brain. So like cognitive impairment, dementia, neuromuscular disorder. I mean, some of this, they're not covering all the problems, but these are some of the problems that a lot of the patients with long COVID are presenting, actually. And it's not surprising that a high proportion of patients with long COVID present neuro-COVID, like neurological symptoms of COVID. I think it's around 70 to 80% of patients with long COVID have a type of neurological problem as well. So this is big, and we need to understand what's going on, right? So I'm kind of like, I think I'm getting to the end. I'm talking too much. I just want to talk really briefly what I'm doing right now, because we developed this monkey model, we published in 2022. And one of the things, because my background is in Alzheimer's disease and neurodegeneration, so one of the things I'm doing now is just trying to see how COVID infection can make it worse, the pathology, how COVID infection can overlap with Alzheimer pathology and and at least accelerate Alzheimer pathology in the aging brain. And this is what we're seeing actually. So when I'm comparing aged, I'm looking at aged animals, non-infected versus infected. And this is a blood vessel image, two images of blood vessels in the brain. And in the left is a control one. So we have the nuclei of the cells in dark blue. And you see like one blood vessel here intact. And when we have infection, not only we have the viral presence, so you see the virus presence inside the blood vessels, but also infecting the surrounding cells, but we also see the structure of the vessel again is compromised. So what we think is happening really is that the presence of the virus is taking access of the brain and is actually accelerating some of the pathology we see in Alzheimer's disease. So we kind of like see the presence of amyloid fibro, which is one of the things we see in the brain with aging. And it's kind of like having this leaky, again, this idea that the blood vessels are compromised. So we kind of like see this leaky blood-brain barrier. And it's kind of like the presence of amyloid, because amyloid is present in the brain of aging, not necessarily Alzheimer, but the presence combined of amyloid with virus can accelerate some of the pathology and can shift patients that are like normally cognitive, like normal cognition, transitioning to Alzheimer's disease because after SARS-CoV infection. And these are like some very, very recent images. I hope we are going to get this as a preprint soon. But you can see here, basically in these images, like you have the neurons here in orange, nuclei of the cells in dark blue, and we see amyloid fiber here. And you see how like the SARS-CoV cells are coming from the blood vessels. So you have like some cells here being attracted by the presence of amyloid. And you see a SARS-CoV cell arriving here. We don't know the identity of this cell. We know from other stainings that, in this one it doesn't show, but from others we know are mostly neutrophils, macrophages, but mostly neutrophils. And you see like this neuron is basically missing a piece here, which I think we don't have the identification in this image, but I think is a microglia. So basically I think something's the presence of this cell positive for spike protein is attacking, is making this neuron die, and or like the presence of amyloid is just attracting more infected cells to the brain. And this is not a coincidence or a surprise. Like there is this tendency, like there is this study that came out less than 2 years ago, just showing that a lot of patients having viral infections increase your chances for neurodegenerative diseases. So in this study, they looked for more than 400,000 patients, and they see like the most association between viral infection and neurodegeneration. So this study came out, they say they didn't include COVID yet because they were going to make a different separate study just for COVID. But you can see other types of viruses are associated with different neurodegenerative diseases. So it'll be interesting to see just COVID, how it associates. But there are some data now coming that is showing that there is an increase in Alzheimer's disease cases. Okay, and future directions, I think that's my last slide. And this, like future directions, how we are going to solve the problem of neuroCOVID and COVID coming to the brain, and how can we help the patients that have persistence of virus in the brain. So first of all, the literature, the data is pointing to viral reservoir, like active viral replication happening in the brain, even after you resolve the pathology in the lungs or pathology in other regions. So that's the thing, like a lot of people like, oh, I'm not coughing anymore. I don't have any respiratory distress anymore. So my COVID infection is resolved. Well, you don't know if you still have virus in the brain. And apparently persistence of viral presence, persistence in different parts of the body are contributing to some of the symptoms that long COVID patient exhibit. And very recent data, and this is such a cool study, just came out like a few days ago, but it's just showing that they basically develop a marker to see spike protein, track spike protein by imaging. And they're seeing like animals can have spike protein up to 3 months. They look at 3 months, they saw in the lung and in the brain. And other studies here in the right is just showing like you have traces, you have in different parts of the body. So like not only in the brain, but in other parts of the body, in the lungs and esophagus, spleen, you can have this persistent viral replication or persistence of viral particles. And this can contribute to some of the symptoms that patients are exhibiting. And that's my last slide. But it's just like, it's this idea like COVID, as I said, coronaviruses can enter the brain. It's not only SARS-CoV, but COVID like basically changed our perspective because there are so many cases around the world. So we never had so many people having this problem of COVID and neuroCOVID. And this idea that long COVID is not, I mentioned viral reservoir, viral remnants, but we can also have different components that the scientists are trying to see. So there is data showing to long COVID and neuroCOVID can be caused by autoimmunity, dysbiosis, tissue damage, and this can all contribute to post-acute viral illnesses. And like so many different things can contribute to neurological damage associated with COVID. So I was talking a lot about direct viral infection, but it's only in blood-brain barrier disruption. These are like the two topics that I talked most, but they're not the only ones, right? I mean, as I said, the brain is really connected to the body. The idea that brain is isolated and protected, it's not so much anymore. So we know like even metabolic disturbances, gut microbiome disturbances can also affect the brain. Like circulating cytokines from the lung can damage the brain, can induce inflammation in the brain. So there is this change in the field now, and we are trying like, okay, how do we prepare? I don't know. But what I know is that we should be answering and should be pursuing these points, which I think are the major important ones for COVID. And I already spoke too much. Time to hear some of the clinical stuff with Dave now. Thank you.
Thank you so much, Danielle. That's great.
Thank you.
Awesome.
So I should share my screen. One moment. Here we go. I'll just gotta master the art of this in a second. So I will share post-disabled screen sharing. Can you share
Yes,
my-
let me see if I can make you
Thank you.
co-host.
Thank you. Now, co-host, there you go.
While David is
so-
setting up, let me just thank Danielle. It's
Yes.
outstanding presentation. Thank you so much. And I have to say, it's amazing research to be able to see how the cells are behaving under these circumstances. And, you know, we know at the macro level about the damage, but seeing directly what's happening in a particular cell is just outstanding accomplishment. Thank you so much.
Fascinating.
Thank you. Yeah, no, it's something we need to keep looking for the like the macro and the micro picture together, right? Exactly like how we're seeing at the microscopic level. There is an explanation for the patients are experiencing. Like they're not going crazy when they say they have changes in behavior. They're like even anxiety, depression can be caused by these neurons being infected. And this is so it's not psychological. People are not fantasizing. There is a neurological basis like of everything that is happening, right? And we need to keep looking for these questions really.
Oh, you're muted, Yanir.
Just thank you both for the outstanding research and also the outstanding presentation.
you.
Really.
I haven't done anything yet.
Hi, David.
We have high hopes,
I'll
David.
start.
Yeah,
No, I'll
wait,
start.
you're gonna see the
I'll
best
get on with it.
now. The best is yet to come. Exactly.
So this is a talk that I usually give to patients, and I always use Danny's picture to demonstrate that there are at least 8 ways that we know of so far that this thing will eat your brain. Firstly, direct neuronal death. The second is more complicated but hugely important. It's glymphatic obstruction. The lymph system, so the body has a sewage system called the lymphatic system. Lymph nodes, spleen, but the brain doesn't. But it has a much more sophisticated pathology, or rather sophisticated garbage system, which is really to remove waste products. And in essence, blood comes in through the artery and then gets washed through predominantly glial cells into the venous system and then discharged in that fashion. The glymphatic system is most active when you're asleep, and in particular in slow wave or deep sleep, where we now know noradrenaline pulsates through these vessels of the subarachnoid to help pump this system through. And so we know that lymphatic obstruction, largely due to the mechanisms that Danny has already explained to you, is disrupted. The vessels themselves are also obviously leaky and disrupted. The Germans have identified at least 21 novel neural antibodies from lumbar puncture samples. It was really the basis for the Berlin Cure 007 study, which unfortunately didn't pan out to be much. But many of these, autoantibodies are directed against specific proteins, and one in particular that selects out for narcolepsy, just saying as a sleep physician. As Danny's alluded to, the blood-brain barrier is clearly leaky. This is a study from acute infection, but the same, I think, is true for chronic infection, as Danny's shown. Even some time later, the vessels remain damaged and porous. And so if you have damaged I can't see if you can see my pointer, but if you have damaged the integrity of this vessel, then everything that shouldn't be there leaks out. Sorry, it's a bit early in the morning here. This is, for me, a humongously important observation and study, because it kind of marries with my long-standing fascination with dopamine. There is a wonderful study, and I won't bore you with it, that demonstrates that COVID causes dopaminergic cells both to die, but also to become senescent. In other words, the pathways that maintain their integrity are damaged, and so they age prematurely and obviously perish. Lots of diseases, from PTSD to major depression, narcolepsy, restless legs, REM behavior disorder, and Parkinson's disease are obviously all features of dopa injury. The one in particular that interests me as a sleep physician is REM behavior disorder. This is the condition that I have long known about and has been described predominantly in men over the age of 50. The caveat to that is that if you follow them long enough, 80% of them will develop either Parkinson's or Lewy body dementia at 14 years. We're not at 14 years yet. So I'll just get you to bear that in mind for the moment. We certainly know that there's only so much lithium to go around. And so when your mitochondria have been hijacked by the virus, whether it is persistent or whether it is still replicating, it is basically turning your mitochondria from being thermonuclear to batteries. And I'm not going to mention the Tesla brand, but in fact, that's what you become. And so if you exert yourself too much, your brain shuts down. If you do too much with your brain, your PEMS gets worse. This is something that just about every long COVID patient will tell you, particularly those with neuroCOVID and bad PEMS. Danny alluded to the gut-brain biome. We know that this virus is actually a bacteriophage. So Carlo Brogna from the Italian group has identified this. And of course, the reason for that is there are more bugs in your bowel than cells in your body. And so it's looking for mitochondria. It's looking for energy and protein to replicate itself. It doesn't come with any of those bits and pieces. And so the most fantastic natural reservoir is, of course, the bowel. And we know there's plenty of gut dysbiosis. The fact is that it actually chooses to eat the good guys and leave the bad guys, which are often facultative anaerobes and other nasty bugs. And so if you've got a leaky bowel, which we certainly know people have for two reasons, one is MCAS, the other is it actually targets the cystic fibrosis translocation receptor gene. Very clever if you're a virus, because it basically makes this membrane more leaky, which is something that we see, particularly in neonates born with cystic fibrosis, is they often present with fecal impaction. This one really does get my attention, and it really speaks a lot to what Danny was saying. So having written a lot about endotheliitis with Razia Pretorius and Doug Kell. This has always fascinated me. It picks out the frontal and temporal lobes. As Danny alluded to, this is where executive function, attention, concentration, and short-term memory live. But in particular, the temporal lobe is about geospatial things. So using the wrong word, not being able to find a word, getting lost driving around your own suburb, having to, use memory aids all the time, putting your keys somewhere and not being able to find them. These are common things that patients tell me all the time. And this is probably one of the features. I would say, just coming back to Danny's observation about the prefrontal cortex, we know that within that area, there is a particular part of the pre-nucleus, which is hugely important in that it actually sends a message to your frontal lobes to go to sleep. And so much of the insomnia may lie in that pathological pathway, which is that you've lost the pre-nucleus, the inability for your prefrontal lobe to tell your frontal lobe it's time to put your lights out. So this is what Danny wanted me to talk about, because this is a talk I've given 1,000 times, but this is kind of how I see things as a clinician. It brings together my years of observational experience. This is the most primitive part of your brain. This is your medulla. Above it is the pons, below it is your spinal cord. Running up the center is the reticular activating system. That is your center of consciousness. That is what we remove chemically when we give you a general anesthetic. That is where coma resides. On one side is the center for REM sleep. You are either in REM sleep or you are awake. There is no in between. REM sleep is entirely serotonergic. And in REM sleep, Sigmund Freud thought it was to prevent, basically protect us from the violence of our dreams. We become paralyzed in our voluntary muscles. So your heart still beats, your gut still works, those things still happen, you still breathe. But anything over which you have complete voluntary control becomes flaccid. On the other side is the locus ceruleus, or the blue body. The reason it's blue is because it's rich in copper, and copper is the main substrate for noradrenaline. This is your flight and fight center. We've long known that when the substantia nigra disappears, that's Parkinson's disease. But in fact, the Israelis some time ago identified that the pathway began down here, and that this may in fact merely be an endpoint for the pathophysiology. Not only did they identify the pathway, they've also done some pharmacology in that area. But at the same time, there's a branch that goes to the amygdala, which is exactly where Danny was seeing pathology. The AMG or AMY that was on her image, yeah, that's in your entorhinal cortex. And it is crucial for memory, but a different type of memory, bad memory. The reason is, I've spent 30 years seeing veterans. When a car backfires, they don't stand there thinking, is that an AK-47 in Kandahar? They literally will fall to the ground. And the reason for that is this is a pathway of survival. It is a survival advantage. To put it bluntly, if you are lying in a cave, and in fact REM sleep is a mammalian land adaptation, if you're lying in a cave and a bear roars, you want to go from being in REM to being awake. You definitely do not want to be paralyzed, and you want to be fully sentient. You also want to fire this guy off so that you will fill your muscles with blood, tighten your sphincters, and run the hell out of there. Parkinson's, I guess, is, nature's way of taking care of that and it becoming barefoot. But the reason we send a message here is a negative message. So bluntly put, you will never go back to that cave. That is why you put that down there on the motherboard. We also know that if you give primates red berries, they'd rather starve to death than eat red berries again, because that's called learning. And it's a negatively enforced learning, and it's encoded with dopamine. All of these things to which I alluded live down there, from restless legs, which we treat with dopa analogues, to Parkinson's disease, which we treat with dopamine. There's a considerably greater increased risk of everything from narcolepsy to REM behavior disorder to Parkinson's disease in PTSD patients. I know because I've studied that. Narcolepsy lives there. I'm seeing more narcolepsy than I can recall. Major depression lives there. So when people tell you that your long COVID is something you should just get over, no, it's true. It is both traumatic and also major depression. REM behavior disorder, what I was alluding to earlier, where people lose the paralysis of dream sleep and will often act out the violence of their dreams. Sleep disturbance. I'll whip through this pretty quickly. It's important for people to realize that, in fact, melatonin is really a product of serotonin. And so, when your serotonin is low because your mast cells are eating all your tryptophan and your serotonin is being depleted, you don't make as much melatonin. That's a key function to appreciate. We recognize that there is insomnia, social jet lag, poor dream content, and flashbacks. People have terrible memories of their times in ICU, all that sort of stuff. But it's also real to recognize that when you go from being super mum, running banks, doing all those things to suddenly being bedbound, there's a considerable unrecognized burden of trauma because you have completely lost your autonomy, and we do poorly not to recognize this in people. I think it's a much bigger tip of the iceberg than we know. Anxiety and depression. Well, as I said, this is all neurobiology. It's not because you need to just suck it up and slap yourself around and get yourself together. We recognize that there are behavioral issues, circadian disorders. We're seeing more sleep apnea. The virus itself promotes obesity and inflammation. And we know that there's damage to the wakefulness circuitry, which is causing more narcolepsy. And so when you look across large populations, what you see is markedly more daytime sleepiness. So that means you need a sleep study. Is it apnea? Is it something else? Fatigue scores, excessive quality of sleep scores, the excessive tendency to fall asleep, all markers of, in my mind, dopaminergic injury and a kind of narcolepsy type spectrum disorder. We know that the glymphatic flow is really important. But in those who've had a COVID infection, whether they were hugely symptomatic or not, in self-reporting, excessive sleepiness, hypersomnolence, is 2 to 3 times greater than controls. Online survey, not a bad one. This is a really quite interesting thing called the Ruse-Sated scale. Are you satisfied? And it's really looking at, post-COVID sleep health. And what they saw was before COVID and now, sleep health is clearly significantly diminished. I won't go into the details of it, but the one thing I would point out there with the red marker is that COVID symptom severity and quality of life as you are at the moment are considerable signals for the fact that sleep is crucial and that people are miserable without it. By the time you're 70, you'll have been asleep for 28 years. There is nothing that you do as much as sleep. There is nothing that is as crucial for the human brain as sleep. And I would dearly love to stick some electrodes into Danny's patients, or shall I say monkeys, to study their sleep and their sleep rhythm disturbance, but that may have to wait. So poor sleep, big problem in individuals who've had COVID. More symptoms at the time of infection, more severe sleep dysregulation. And that, this is something that I've been saying, only for 4 or 5 years. Sleep health should really be looked at in those who've had COVID. And we should look really at both their physical and mental health well-being. I'll skip this one because it's a little bit boring. But I will point out to you that obstructive sleep apnea, which is certainly a very prevalent disease. Excuse me, I just needed some water, folks. Is associated independently with worse outcomes for those who may have sleep apnea, which is not merely obesity. It can be craniofacial, it can be familial, it can be, there are certain ethnicity groups to whom apnea is a greater risk. It is an independent risk factor for developing long COVID. Again, another reason to screen people who've got long COVID for sleep apnea. I would also say that in a previous life, well, 25 years ago when I was still writing science. I never thought I'd come back to it at this point in my life, but my PhD was in the neurocognitive impacts of obstructive sleep apnea when everyone was more interested in blood pressure and heart disease. And really, it was very obvious to myself and a couple of others who were interested in the area at the time that attention, concentration, short-term memory, daytime sleepiness, grumpiness, irritability, motor vehicle accidents, short-temperedness, and forgetfulness are all major features of obstructive sleep apnea. And so you want to know whether people have got an additional burden of this condition. It's an independent risk of PASC from the Recover study. And the one thing we haven't seen is that this isn't present in kids. So it may be coming back to what Danny was saying about what we call frontalization. So your glial cells actually prune your brain from the back to the front. So we do our frontal lobes and maturity last of all. And so that's why, certainly in Australia, women frontalize faster. So they usually are done by 22. Men are not done until 25, which is why I had to pay an excess on my insurance for my boys to drive until they were 25, because we recognize that youth and stupidity don't really change until blokes get to 25. Some may argue we never get there, but I'll leave that with you. But in truth, 25 is the magic number where you decide that, maybe heights aren't so good for you. This is an Australian study. And what I wanted to point out was the potential for the effectiveness of novel orexin receptor antagonists. What we're talking about here is a drug, an agent called modafinil or Nuvigil, its later entity, which is a wakefulness promoter. It is a uniquely dopaminergic drug. It was, in fact, first discovered by a French company who'd never made anything useful. They were trying to make a dopaminergic antidepressant, and it never worked. Their only saving grace was that the French Foreign Legion read widely and deeply, and so they used it in their Foreign Legion guys who were in the First Gulf War. They found it to be fantastic to for keeping soldiers alert. And so it was that accident that made them billions of dollars. And it is a fascinating drug, and certainly one that I am increasingly, I always use it in narcolepsy, but in the heavily somnolent people with neuroCOVID, there's clearly going to be a role for this. And again, it is specifically a dopa agent, as I keep saying, my dopa fixation. Our race to the bottom. So, Australia was the last to open. And so you can see that, during our periods of lockdown and other things, there was abnormality. But there's absolutely no question that our total sleep time really started to accelerate downwards. With narcolepsy, again, as I indicated at the outset, there is a specific autoantibody that codes specifically with these long COVID autoantibodies. And so, as I keep saying, we're going to see more of these people. But it's also important to recognize that these autoantibodies also interact with complex regional pain syndrome and, as I said, narcolepsy type 1, and in patients with small fiber diseases. So as Danny alluded to, it's not just the brain, it's the entire nervous system. And we are seeing a lot of people with new onset small fiber neuropathy, and that alone is concerning. We know that restless legs, again, low dopamine, is associated with long COVID in women. In those that have been studied with long COVID, The similarity, so just excluding sleep apnea, the similarity between those who have long COVID and those who are chronic insomniacs is that what you see is this reduction in slow wave sleep. And I hate to come back and circle back to what I'd said at the beginning, but it is slow wave sleep that drives the brain to clean itself with the noradrenergic system. And so when you look at those who have insomnia as part of their long COVID, they are almost certainly going to have issues with glymphatic drainage, and that is in itself an issue. Just to finish, we are starting to see, this is the first paper where they saw 4 patients who had REM sleep without atonia. In other words, they had lost the paralysis of dream sleep. And the question that they asked is this a sign of CNS pathology? Well, yes. Bottom line is it is. This is from 2021. The dopamine paper didn't come out until 2023. So we were already seeing the signal. I was already watching this literature. And so I got my sleep fellow last year just to do something interesting, which was to collect all the sleep studies on these people. Now, I will tell you, we excluded those who have narcolepsy because there's a 30% overlap. But what you'll see is it's males and females. Their mean age is not, over 50. Many of them you will see here are quite young, and we've included a 17-year-old. I also have a 16-year-old. And since the beginning of this year, at least 5 others who are under 40. The striking thing to point out, though, is just how much of them have fatigue and brain fog. 75% of them present predominantly with neuroCOVID. And these people all have REM sleep without atonia on what we call a SynBar montage. We add extra electrodes to voluntary muscles, and that's the best way for us to make this diagnosis of REM sleep without atonia. Some will actually have visible movements, which is REM behavior disorder. And so the big question for me is how much dopa damage are we doing? And are we going to see more of these things in time? So I will stop sharing. I'm done. Thank you for having me.
Hey, so it was definitely not sleep-inducing, that presentation. And as always, I think that one should say it was a great presentation, but also thank you for the stand-up part of it.
Absolutely. Would you like to take a few questions, David?
Yes, sure. By all means, happy to.
Awesome. Okay, let's see. Why don't I take a look in the chat? There's been quite a few, so let's see if I can call on somebody and have them ask their question. Louise D, would you like to unmute and ask your question? If Louise isn't available, maybe we can ask Aurora Horsecamp to come on.
Yes, I was wondering about the use of melatonin for patients who have sleep issues post-COVID and any particular dose or brand
Yep.
since they're not FDA regulated. Thank you.
Yeah, that's a good question. Yes, I use it in all long COVID people. My preference is a pharmaceutical grade component, compound, well, not compounded, but a pharmaceutical Therapeutics Goods Administration certified. So for us, it's FDA approved. It requires a prescription. It's called Circadin. It's a slow release melatonin, and it's 2 milligrams. The idea that some is good, more is better isn't the case. At 2 milligrams, there is some endothelial protection, and as I said, we've written a lot about endotheliitis, and Doug and Razier have clearly shown microclotting and other things. The reason I use the melatonin slow release is because it's reliable. and it's going to come on if you take it at 7 p.m. at 9 p.m. The merit of that is that that is your natural circadian rhythmicity, which is lost in patients with long COVID. And every hormone and every enzyme in your body needs to know what time of day it is. So alcohol dehydrogenase, which breaks down alcohol, comes on at night. That's why if you drink during the day, you will get more intoxicated. HMG-CoA, which is the enzyme that breaks down cholesterol, switches on at night. That's why the original statins were given at night. And your cortisol bottoms out at 3 o'clock in the morning. Now, your adrenals and liver can't see what time of day it is. They rely on the circadian rhythm to basically take their cue. It's the atomic clock of your body. And so, yeah, that's my preference is slow release 2 milligrams, 7 or 8 p.m., no later.
Excellent. Thank you. Let's see. There's another question from Bree. Bree, would you like to come on, Bree Street, and ask your question?
Oh, hi. Thank you very much. Fascinating talk. Really appreciated it. I was curious, you mentioned the norepinephrine pulses in glymphatic movement and clearance, and
Yep.
I'm wondering about clonidine and those types of medications. Do they impact that potentially?
Look, to be honest, I don't think anybody has really looked, and I haven't seen much data. This is pretty, much of the glymphatic mechanisms have really, we only started to even know this existed about 11 years ago. Danny might have a better vintage of that than me, but it's novel. And so a great deal about how it works, and the noradrenaline pulsing, that's a publication from only a few months ago. So if you're reading everything, you kind of see stuff, but I don't know. No one's really looked at the pharmacological mechanisms. The biggest problem may also be that if you increase cerebral blood flow through already damaged vessels, that's something that always focuses my mind, is that hypertension is not your friend. And we already know from the flow-mediated dilatation stuff that I wrote with Razor and Doug, that new onset hypertension is a big problem post-COVID because of the vasculitis, the vessels are stiff. And so there is, an increased risk of hypertension. The vessels just don't relax normally.
Such a tough disease.
Yep. My 40th year in medicine, I have to tell you, I haven't seen anything tougher.
Yeah, you mentioned something about amyloid, the creation of amyloid on the brain due to COVID-19. Is the pro-BNP test can determine that?
I'll give that one to Danny.
Sorry, I understood like the amyloid, the presence of amyloid and COVID, and what's the other question? Sorry.
It affects the brain, right?
Mm-hmm.
So is that a part of COVID-19 long COVID effect?
Yeah, so
And the
that's
function, the heart wouldn't function well. Yeah.
so in terms of like amyloid, I can talk about like 3 days about this, but just to summarize, the idea is that like amyloid, and it's one of the things we see in patients with Alzheimer's disease, is actually accumulation of some proteins that we call misfolded proteins. So amyloid will be like a misfolded protein, which is basically, like, to summarize, is a protein that shifts the structure. Instead of having a normal structure, this protein shifts for reasons unknown mostly, and it kind of like start accumulating in the brain, and this not only in the brain, it accumulates in the body as well, but in the brain can cause some of the damage we see in Alzheimer's disease. But the thing about amyloid, which is interesting, is that we humans and monkeys as well, like it's a primate thing, we accumulate amyloid in the brain with aging, and not necessarily having amyloid in the brain can mean you're going to have Alzheimer's disease, but it can contribute. And what we are seeing in the lab, but other labs are seeing as well, is that there is this correlation, like the presence of SARS-CoV in the brain can accelerate amyloid pathology in the brain. So this idea that if you have the viruses and the virus is changing how the cells are interacting with each other in the brain and how you have inflammatory response. So the fact that you're having a virus in the brain can induce this protein to accumulate in the brain and accelerate. So this idea that it is, again, like we're just 5 years from the pandemic, right? So it's something that we can barely have a dimension, because so it's really hard to estimate yet how much of the people are going to have memory problems or cognitive problems or Alzheimer-like memory decline. We cannot even say we're expanding. This is one of the things, like the scientists are expanding the because of COVID, because of neuroCOVID, we're starting to say, instead of Alzheimer's disease, we're saying Alzheimer's disease and related dementias. This is how we have to write grants now if we want to ask for government money. We cannot say Alzheimer's disease anymore because there are so many conditions that are like Alzheimer-related dementias, Alzheimer-like, and COVID can be one Alzheimer-like. So it looks like, and we cannot measure yet, but it looks like we're going to have a lot of people with cognitive problems. And one of the things, it's because you have this increase of the amyloid production in the brain caused by the viral presence. And we are quantifying this nowadays with patients that already had mild cognitive impairment. So for older people that already have some type of cognitive problem, we already know they're accelerating and developing Alzheimer's fast. But we don't know for the rest of the population because we're just 5 years on that.
So it will be classified like a comorbid condition, right? COVID-19 and the effect of COVID-19 on the amyloid or whatever. So it's like a comorbid condition.
Yeah, it can be like a contributing factor for the pathology. Yeah, can be. Yes.
Okay. Thank you. Shokantala, let's give some other people a chance to ask questions. Ifra, would you like to come on and ask your question?
Yeah, sure. First of all, thank you both for your presentations. It was really fascinating to learn about your research. My question is, and either of you can answer, but have you noticed any efficacy on like low dose naltrexone for long COVID patients and, managing symptoms such as fibromyalgia or chronic pain?
That's more for you, Dave.
Yeah. Look, the original data in ME CFS was a single paper with 39 individuals who were given low dose naltrexone. And, the outcomes were not objective, they were subjective. And so I am not entirely sure. What I do know is that we use naltrexone to block opioid receptors. And so, for some people that have tried it and, in the land of desperation, and I completely understand that, I've seen many on it. And it really cranks up their vascular headaches and migraines. So that often subsides when you stop the naltrexone. For those who think it's doing something, I don't care. I'll leave them on it. But often in those who stop it, there is no demonstrable decline in what I see. It's hugely expensive. So, I would rather that they were spending some of that money on the agents that we know are particularly useful for mitochondrial support. And oxaloacetate is probably the heavy lifter and is certainly the most expensive. So, it's in Australia, it has to be, made up by the pharmacist, et cetera. It's really expensive. And for people who can't work, for people who are doing limited work, for people who are really, struggling financially, it's a big burden for something. There is a big study being done, and like everything, I'll wait to see what the science shows. But the original theory behind it was based on a single small study, and so perhaps I'm a little less convinced.
Okay, thank you so much.
It's a pleasure.
Sharon, sorry, I was just going to say,
ahead.
sorry, Sharon, sorry to
That's
interrupt.
okay,
No,
go
I was
ahead.
There is a question here that I think is really good. I think it's a good one to answer. It's from Louise, and it got some likes. She asked, like, why are so many patients psychologized for symptoms with little to no test scans done? Which test scans should be done on patients before a diagnosis of mental illness is given? And I just want to touch, like, and because I think it's such an important question, and if Dave wants to jump in too. But one of the things we are doing now with the long COVID advisory group is just writing a guide for physicians and healthcare people, just like which type of exam should be asking. And I know for a fact that neurologists have been having I know has been hard for patients and the neurologists to understand, like, okay, before a diagnosis of you're too anxious, is the lockdown made you anxious, the lockdown made you depressed. Like, there are exams they can ask. Like, I know they usually don't ask, like, especially imaging exams like MRI and PET scan. So, like, things you can see if there is different densities in parts of the brain, if there are areas with focal inflammation. These exams are expensive, so I know there's not a routine to ask them, but there are exams available. There is, like, not only imaging, which is very, very expensive, but even like blood exams. That are blood exams nowadays that you can detect neuronal damage. So basically, if you have neurons dying inside your brain, they release some type of protein that goes to your blood, and doctors, neurologists, and other medical doctors can ask for this type of, if there is a suspicion that something wrong. So there are exams that can be asked for, and it's just not being routinely asked. We know it's a complication for the patients, but so, like, neurofilament light, which is the name of exam that we can ask in the blood, MRI for brain atrophy. So there are exams that should be requested before, just say, depression, anxiety. And even depression, anxiety can be caused by this dysfunctions and structural changes in the brain too. So it's something you have to keep fighting for, right, Dave? Like, I bet you.
I absolutely. I think that was my point about trauma and anxiety. They're neurobiological. And we know there's direct neuronal injury in the areas that drive everything from impulse control to mood. We know that there's reduced serotonin. We know there's low dopamine. These are not strangers. I do an MRI, special sequences that radiologists with whom I'm friends do for me, of long COVID sequences. And, when you start to see 17-year-olds with gliotic lesions in the frontotemporal lobes, that shouldn't be there. And so it's also, I think, important to get a baseline scan, because if you're going to be using nattokinase or anticoagulants, you want to know whether there are microhemorrhages or even new aneurysms. The combination of loss of elastic tissue from MCAS coupled with rising blood pressure, as you demonstrated, there's a perfectly beautiful looking aneurysm in your image of that blood vessel. Those are things that you want to know about before you just whack people onto drugs. So there's that. I think the standard testing, that's one of the things that we emphasize. So I wrote a paper with the British Psychological Society, and they published that. And it was really about why you don't psychologize long COVID. But what I was actually really hoping was to get the psychologists to help with behavioral strategies for the clinical anxiety. So that means people who are on antidepressants, those sorts of things, they also need an additional support. There's lots of non-pharmacological therapies for PTSD, from expressive art therapy to EMDR to dream rehearsal therapy. There are other non-drug things that these people have wonderful expertise in that would be very helpful. But I think it's also, just to finish, really important that that people are not diminished by this. It's biology, it's biochemical, it's pathophysiological. It's just a shame that not everybody reads enough.
Great. Thank you. I think we have time for maybe one more before our time is up. So let's see.
There's a question that's been
Oh,
asked,
yes, go ahead.
and maybe it could be discussed. Of course, we're learning about damage. I mean, we know that people are suffering from all kinds of neurological symptoms. If you look in the brain, you will find the evidence. But one of the things that people would like to know is how much of this will heal over time. And the other question I think that's important to talk about is the consequences of multiple infections. So the role of prevention in subsequent infections.
I'll go. Um, Look, gliosis is an endpoint. That's a dead brain cell. A lot of the stuff that Danny's showing with accumulated proteins, they don't go away. And so, the brain has neuroplasticity. There's no doubt that, the brain, particularly the younger brain, can open new pathways. People can relearn. Again, that's why we need rehab people to, in the same way as we rehabilitate people with stroke to, learn how to speak, walk, talk. So there is neuroplasticity, but damage is damage and dead cells are dead cells. And that's the bottom line in terms of that. But you have to then also appreciate that the virus is still there. It's still in our brains with all these, multiple vascular, gliotic, glymphatic, all of these pathophysiologies are still ongoing. And so, until we can actually work out how to get it out of these reservoirs, I suspect that and it's just my assumption, that there may be for a group, not all, but for a group, they will continue to have problems. So when I see young people who've got new onset REM behavior disorder or narcolepsy, the one thing I warn them is you definitely do not want to get COVID again, because, this is not going to help you. And I think that comes to the question you're asking, Yania, which is repeated infections. We know unequivocally that the risk is cumulative.
Mhm.
Your risk of getting influenza, proper flu, is about once every 8 to 12 years. You can get COVID every 3 years, or 3 times a year, every 4 months. And in fact, with Parolaclan and its increasing immunoevasiveness and the fact it's mutating so quickly, we are seeing people who are getting repeated infections within a year. And so you never recover. And we now know from a paper recently that the immunological changes in B cells and T cells, as Anthony Leonardi rightly pointed out, don't get better in a hurry. It's months, and they're still present out to 6 or 9 months, maybe longer, by which time you've already had another infection. And so there's absolutely no doubt that the risk of long COVID goes up with every infection. I think for me that evidence is pretty stark, and I think that it's pretty real.
Yeah, and just to complement briefly, this idea that like you have this viral reservoir and you can have in the brain and other parts of the body. And like in this paradox, like the virus is able to hijack and access very, very small niches of parts of the body like the brain, but we don't have drugs that do the same, right? So it's like the virus can penetrate to parts of the brain, deep brain, that we don't have therapies. We don't know how to do that because our blood vessels, our blood-brain barrier, it filters that. So it's like, and it increases the chances of getting more viral reservoir and getting long COVID increases in each infection you have. It's like a lot. So it's like prevention is really the key for avoiding damage and neurological damage as well. And this, and like as Dave was saying, like the brain is plastic. So in terms of like the cellular pathology, you can have recover. But at the same time, we have to realize that neurons are non-renewable cells. So the neurons we have now are the neurons that basically we were born with. And it's like, so it's like once they are gone, they're gone. There's not a lot we can do. We have newborn neurons in very, very specific, less than 2% of the brain, adult brain, basically. So we don't know how to generate new neurons to replace the ones that are gone. And if you have a virus that infects neurons, it's a major concern, right? Like, how do you avoid that? So it's just like, but we have new inflammation that responds very well to plasticity. We have other, so we can readjust, we can redo our circuitry in the brain, even if you have neurons, some neurons dying. But it's a difficult process. And that's, Dave said, like, we need people jumping in recovery. We need specific protocols for, like, how do you treat people with neurological damage like this? And how do we find a way, maybe it's not going to be the same behavior as before, but maybe like find a solution to like a pathway of improvement, because improvement is possible. It is just really hard. And it's something you really want to avoid, really, especially reinfection. Yeah.
Thank you.
Yes. Thank you so much. These have been great presentations, great questions and answers. I think our time is about up. So really appreciate all the information and the time you've given us. If anybody has any questions, you can reach out to me, Sharon, at necsi.edu is my email. We will have a recording available soon on the WHN YouTube. And to learn more about the WHN World Health Network, go to whn.global. And we really appreciate everybody's time and attention. Have a great weekend. Thank you so much.